Thank you very much. It's a great pleasure to be here today. Monty Python would say, and now for something completely different. So I cannot promise that you'll get anything that you can apply immediately to your daily work, but I hope to provide you with food for thought, perhaps for the hour afterwards, and give you one or two interesting nuggets about ourselves. So I have 3 goals today. There we go. I'll tell you about something called apoptosis, or apoptosis. We can't agree. It's like potato, potato. It's one of these sayings depending where you come from. I'll tell you what this is. I'll explain to you why we have this program in our bodies, and then we'll discuss a little bit at the end the molecular mechanism, but I'll try not to go too deep because that's mostly for molecular biologists aficionados. So let's start with the first question, apoptosis, what it is. The term was termed by these 3 gentlemen here in the 70s of the last century, so not quite 50 years ago, to describe a set of morphological changes that they saw in tissue slices. So these are 3 pathologists. In healthy tissues, every now and then they saw cells that would spontaneously die in tissues, and they always looked the same, and they said, isn't that weird, cells that always die the same way? And so they decided there must be something more fundamental behind that, and called this apoptosis. Today, we use the term to describe what we call an active cell suicide mechanism. So the idea is we are multicellular animals. Our body is a community of cells. You could think of our bodies as little countries if you want. We have a lot of citizens, but each of these citizens, these cells, has much less freedom than we have here in Switzerland. They only exist, their only reason to live is to serve the body, right? And because of that, if a cell has no reason to live anymore, or if it becomes a danger for the organisms, or if generally its death would somehow help the community, this cell will readily commit suicide. All right? So for those of you who have the Swiss history, you can also call this the Winkelried program, right? So the idea is about the Battle of Sempach, the Swiss against the Austrians. Swiss are getting battled badly, and then Winkelried decides to sacrifice himself to help the Swiss win the battle. Winkelried dies, but the Eidgenossenschaft, the Swiss Confederation, flourishes after that. Same thing here, but at the level of individual cells. All right. The term itself, apoptosis, it's Greek. It means to fall off, and so it's a gentle death principle. The Greek used that term to describe the fall of leaves in the fall from deciduous trees. So the same concept, programmed loss. And apparently they also use it to describe the loss of hair from aging men. Voilà. So we're going to use this as active cell suicide. So the concept is relatively easy. We have a cell. This cell somehow realizes that it's not meant to be there anymore. It kills itself, it's suicide, and then it gets removed from the body, mostly by being eaten up by neighbors. We're not going to discuss this part. But basically, the cell that dies emits a signal that said, dear colleagues, dear neighbors, I'm about to die. Please take care of my body. And the neighbors look over and say, oh, free food. And then we'll grab that cell, engulf it, and then degrade it and reuse the contents. All right, so why do we have a program like that? There's lots of good reasons. The most visible reasons, I think, are during development, because you have a lot of very visually pleasing examples of that. Let me tell you perhaps about this, separation of digits. So when we're little embryos in the wombs of our mothers, our hands initially are like ping pong paddles, right? So they're basically just flat surfaces. So where the digits are, the cells will continue to grow and differentiate into bone cells and muscle cells and so forth. But at the beginning, we also have tissue between the digits. And so this needs to go away. And this happens relatively rapidly. You have here this is from a human embryo 48 days after fertilization. You can see this paddle. You see the tissue between the digits that are forming. 3 days later, you can see that much of this tissue has disappeared. What happens? These cells get a signal from the neighbors saying, ladies and gentlemen, you're at the wrong place. Please do the right thing. And so then these cells actively kill themselves and are cleared, so that a little bit later, then you see that you have lost this cleft. This is programmed. And it happens because these cells get that signal at that point in development. And we can show that because sometimes these programs goes awry. One example in humans is what is called simple syndactyly. Sometimes children are born with a defect where the tissue between the fingers simply did not go away. And so then they have tissue stuff between the fingers. This also happens over evolutionary times. For me, the big difference between ducks and chicken is that chicken have a lot of programmed cell deaths between the toes. Ducks don't. Because ducks don't, ducks have palm feet. The tissue between the toes stays and they get palms. So you can see how changing that program can actually be beneficial. Another example of a very useful use of programmed cell death during development is what I call safety in numbers. So you have to realize that building a human body is a very complex endeavor, and you only have one chance of doing it. You can't try to do it and then realize, oh, I screwed up, let me try again, right? And so that means that you need to get the right type of cells at the right place, connected in the right proportions. So let's take a graphical example, the muscle in my big toe here, right? So the muscle in my big toe, which allows me to move my toe, the cells are here in my lower back. So during development, the cells in my lower back need to send an axon, a process down the leg, all the way into the foot, up to the toe, and need to tell those muscles when to contract. Now, how do my cells in the lower back know how many muscle toes will be in the toe? They don't. There's no way to know, right? So what do? We do the same thing as engineers. We put in a fudge factor. We say, well, there'll probably be around 20,000, so you know what? I'll make 40,000 neurons, and I'll let them on their way, right? And then some of them will reach, some of them will go the wrong way, some of us will lose, they'll lose their way at the knee perhaps, I don't know. And then at the end, we look who's connected. So these guys will say, oh, I'm connected, this is good. This guy knows, oh, I reached my goal, it survives. And all the other guys that did not get a connection, they simply disappear. So during the development of our brain, we lose about half the cells. Half of our brain cells we lose before birth. If we didn't have that death, we'd have brains that are twice as big, but they wouldn't be better, right? Because the connections would be all wrong. Now, of course, this doesn't only happen at this level. This cell also, it's not connected, it's not useful. Also, these cells will often then go. And this becomes even more complicated if you think that in the brain you have signals not only from the cells that you innervate, so your targets, you often yourself are the target from other cells. They'll also tell you whether you're okay. You'll get targets from among yourselves. Our cells usually are in groups called nuclei, and if one cell goes the wrong way and is all alone, it has no business there. And so these guys basically keep telling each other, we're happy, we're a team, we'll survive, and if you're all alone, then you simply die by desolation. And finally, there's additional signals here along the axons that simply make sure that the cell knows that it's functional. Over the last 20 years, people have learned a lot about these signals. I'll just give you one example. Think of these as Lego blocks. These are called molecules, proteins. And so this is a family of receptors which have these various survival signals. So the idea is a cell will secrete this signal, would emit a signal, and any cell that has the right receptor will survive. And if this cell has this receptor but doesn't get this signal, it will die. So you're basically addicted to this thing here. You get it, you survive, you're happy. You don't get it, as long as you have this, you're going to kill yourself. So how is this used? Well, one example that I can show you here are the sensory neurons we have. So our skin have many different types of sensors. We can feel temperature, we can feel touch, we can feel pain. We even have some muscles in the hair follicles. You feel your hairs being straightened up. And these different kind of sensory neurons, they're all in the same place in the body, right next to the spinal cord. But they use different receptors and they get different signals. And this ensures that this type of neuron, which is associated with hair follicles, only survives if it goes there. If it goes somewhere else, it dies. The cells that are used for pain get their signals from here. The cells that we have to the muscle that allow us to know, even when I close my eyes, I know where my fingers are. Those are proprioceptive neurons. They survive if they sew up to the muscle spins and so forth. And so this allows you basically to have a mix and match and make sure that the right type of sensors are attached to the right type of things at the end. Development is only the first wave where we use programmed cell deaths. There's a second reason to have this throughout life, and that is to control the number of cells we have and to make sure that we have the right cells. You have to realize that, again, our cells are able to sense their environment, right? So they're able to feel whether the neighbors are there. They're able to feel how much oxygen they get, how much sugar they get, what the temperature is, what the pH of the solution is, and so forth, and so forth. And they get all this information, and at the end, they add it up and they ask themselves, am I still doing okay? And if they're not doing okay, then they say, oh, well, then I should die. And they kill themselves, right? So, and this is really challenging because you can't even imagine. They get a lot of different things. So let's say a cell gets infected by a virus, and the virus starts replicating, and that screws up the system in the cell. The cell realizes, oh, something wrong with me. What does it do? It kills itself before the virus can multiply, and this saves the neighboring cells, because if I kill myself before the virus replicates, then it can't infect the neighboring cells. So this is a little bit like Sigourney Weaver in Aliens 3, I think, right? So she has this this queen alien in her body, and what does she do? So at the end, she jumps into this giant vat of metal, molten metal, something like that. She dies, but the alien queen dies also, and so her colleagues are saved, humanity is saved, at least until Aliens 4. Same thing, a cell that's on the way to become a cancer cell starts dividing, multiplying without getting the right signal saying you should multiply, realizes that, will kill itself before it goes. So this suggests, or this indicates that there must be sensors. The cell must be able to sense what's going on and must be able to add all of these sensors together. And that's one of the big mysteries we'll come out at the end, that for many years we could not understand, how does a cell even sense things, right? These cells, they're like Woody Allen. They must be completely neurotic, going through life going, oh my God, what am I supposed to do, right? He tells me I'm okay, she tells me I'm not okay. And somehow you need to integrate this information and then make a 1 or 0 decision, right? Apoptosis, programmed cell death. You can't have a cell die 20%, right? Either it dies 100% or it doesn't die at all. So you need to have a binary switch integrating a lot of analog signals into a digital output. So another example here where a programmed cell death is useful. Let's say I said cancer-prone cells. Here an example, you go to the sun, it's beautiful sunny, you go on the Utoke, you get a lot of sun, you get a sunburn. The sunburn basically is a whole bunch of cells in your skin dying. Why do they die? They got damage through the UV light in the sun, and this damage, the cell will try initially to repair it, but if you get too much damage, then the cell starts thinking, I'm not sure I can repair all of that. And if I don't repair it properly, I'm going to mutate, and if I mutate, I'm on my way to cancer. And so rather than risking that, I'll simply do the safe thing. I'll kill myself, I can be replaced anyway, I'm just a skin cell, and that's what happens, and that's why we have these sunburns. If we don't have apoptosis, and this can happen either through defects or other processes, then you will have increased mutation and you'll have increased cancer. And indeed, there are patients, there are people that have defects in this apoptosis program, they're prone to cancer. They get much more cancer. In fact, the shocking thing is not how much cancer there is in the human population, the shocking thing is how little there are. If you think, we have 10 to the 14 cells in our body, each of these cells gets 10 to the 5 damages in our DNA every day of our life, over 70 years, let's say. 10 to the 17 times 10 to the 5. Now again, only one person in 3 gets cancer. I don't know. I think it's pretty good. All right. So the corollary of that is, of course, that if you do get a malignant cancer, something probably went wrong. Why didn't the cell kill itself? The answer is that normally either the cell is not able to sense that it's abnormal, there's a defect in the sensing mechanism, or it senses it but it's not able to kill itself because there's some leak or some defect in the system. All right? So most cancer cells have during the development acquired the ability to evade at least partially apoptosis. It's one of what we call the hallmarks of cancer. There are some others. Ironically, in addition to that, although the good cancers are at least partially responsible or partially resistant to apoptosis, most of the classical ways we treat cancer are ways where we damage the cells and we reactivate this killing machinery in the cancer cell. We damage the DNA or we screw up the cell division machinery and so forth. And what happens is that these preferentially kill the cancer cells because often these cells are rapidly dividing, they're multiplying, and these damages do more damage to a cell that divides than to a cell that is quiet. All right? And so this means that if you know how to tweak this, if you could activate apoptosis again, you'd have a blockbuster cancer treatment. Right? So imagine, all these cells in my tumor, in my cancer, they're abnormal. If they suddenly could realize again, oh, we're abnormal, we should be dead, and they kill themselves, the cancer would disappear by itself. Right? So this is the dream, the pipe dream of the cancer biologists working on this apoptosis. All right? And of course, if this was feasible, then you'd have a billion-dollar market right away. This is a very old forecast from 2009, opportunities in cancers and other diseases, and so they forecast a boom from 2008. So it was a small market at the time, a 20-fold increase over 6 years, 5 years. Average growth rate of 64%, and yeah, pretty good. Sounds like good AGRs. The idea was that at the end, about every fifth drug on the market would selectively kill cells using this knowledge. Now, if you look at what happened, we're nowhere there. We're 2016 now. We're not there yet. As is often the case in the life sciences, in the medical field, vastly over-optimistic predictions, not because the basic logic is flawed, but simply it's more complicated than we think. All right? So long-term potential definitely is still there, and I'll show you at the end a few examples, but it's not that trivial. And the reason it's not that trivial is because, again, yes, there's ways to kill a cancer cell. In fact, there's no cancer you cannot kill. There are only cancers that you need such a high dose that you not only kill the cancer, but you kill the normal cells in the body and the patient dies, right? And so the challenge is that selectively inducing apoptosis in cancer cells is difficult. Non-selectively inducing apoptosis in any cell is easy. Right? You just damage the cell goes, oh, something's wrong with me, kills itself, it's dead, right? And of course, that was generated. This is a very, very, very long list. It was completed in 10 years ago, 20 years ago. We stopped putting new things on the list because basically everything in the kitchen sink, you throw it on a cell and it will induce apoptosis at the right dose, right? So already Paracelsus says the doses makes the poison. Anything at high enough dose will screw up the cell. The cell realizes, oh, something's wrong with me, kills itself, and it's gone. All right. So now because the fact that a cell that feels abnormal kills itself, and this is used to protect ourselves, you'll find apoptosis involved not only in cancer, as I mentioned, but in many other diseases as well. I explained cancer. Cancer cells should be abnormal, should die. If it doesn't, you get cancer. Autoimmune diseases. Immune cells that recognize their own cells should be eliminated during their development. If something goes wrong and they survive, they'll attack our own body. It's an apoptosis defect. Sometimes the opposite happens. Let's take an example, stroke, right? So during a short time in my head, in my brain, one of the blood vessels clogs, and for a time, the nerve cells in my brain don't get enough oxygen and sugar and what else. The cells at the center of this insult will die relatively rapidly. We'll come to that, using something called necrosis, right? They don't get enough food, they don't get enough oxygen, they simply die. The cells at the periphery, in what is known as the penumbra, they get less oxygen, they get less food, not enough to die, but enough to be mighty uncomfortable. These cells go and say, whoa, that was weird, what was that? Oh, maybe something's wrong with me, maybe I should die. And 24 hours after the stroke, they actually kill themselves. Not because they're not useful anymore, but simply because they had this insult. And in animal experiments, if you block apoptosis in these animals on the 2 days after the stroke, you save half the cells in the stroke. And you can imagine that makes for a much better prognostic, much less damage in the brain. Same things for a heart infarct. Just replace brain with heart. Hepatic disease, I'm not going to go through all the examples. But you see, sometimes cells that we want to keep die because they think they should die. Sometimes cells that we would like to get rid of don't die. And so you can play the system both ways. All right, so let's summarize what I told you so far. Apoptosis, active cell suicide mechanism used by multicellular animals, so people like us, to remove cells that are either in excess, in the way, or potentially dangerous, right? So in excess or in the way in development, potentially dangerous anytime. Defects in apoptosis are observed in many human diseases. Sometimes there's too much of it, sometimes there's not enough. Now, apoptosis is just one way for cells to die, and that's important to know. There's also many other ways. I already mentioned necrosis. So what's the difference between the two? Apoptosis, I told you, is programmed cell death. So the cell has a program, an internal program, that allows it to basically shut itself off, right? So it kills itself, it gets removed. Necrosis is what we would call unprogrammed cell death, right? So what happens here is you have a cell, something really nasty happens to it. It simply cannot survive. It will also look funny, but funny in a different way. And it will also then die. Normally in necrosis, what happens is that the cell, rather than being cleanly eaten up, it will simply go splat. It will leak and the contents of the cell will go all over the place, and that will lead to an inflammation. So to give you an example based on here, computer, right? So there's a computer automatic program shutdown. So I have here shutdown. If I click this button, the computer shuts down. Activates a program, the computer shuts down. Alternatively, I can open the window, I can take them out, and I can throw it out. Unprogrammed computer shutdown. By the time it's on the floor, on the street, it's off, right? But it's messy, right? So this is apoptotic computer shutdown. This is necrotic computer shutdown. And these are just one example of both types. It turns out that there's other ways to have a programmed cell death different from the system I'm telling you now. We'll talk about the molecular biology, the basis underlying that in a few minutes. And there's also many different types of unprogrammed cell deaths. In fact, sometimes, and then it gets really confusing, you get things that are programmed but look unprogrammed. So there's something called necroptosis, which turns out to be unprogrammed cell deaths, right? No, programmed necrosis. So this looks like it's unprogrammed, but it's actually programmed. Here's an example of that. You can recognize the gentleman. This guy will necrose, right? It's going to be, but it's programmed. We know exactly how it's going to happen. We can't tell exactly when, within a few minutes, but this is what happens. All right. Now, why do I belabor this point? Well, it's important for us to know. Remember the first slide with these three pathologists, right? So we want to know how a cell has died. I have a patient. He or she has a disease. I see that cells die. I'd like to know why. There's a good reason to ask the question why. Two reasons are mostly different types of cell deaths have different meanings. If a cell dies by necrosis, if I cut myself, the cells at the beginning will die by necrosis. It will have a different meaning, not only for me as a physician, but also for the cell, for the body. Necrosis means bad thing, right? I cut myself, it's unprogrammed, it's nasty, and I'll get an inflammation. That's why it gets red and hot. Programmed cell death, apoptosis, is not nasty. It's wanted, it's desired. And so in fact, necrosis is inflammatory, activates inflammation. Programmed cell death, apoptosis, is anti-inflammatory. The second reason we want to know why or how a cell dies is because if I'm going to think of using a therapy, I need to know what mechanism it is, right? And so here the challenge, of course, is then to know, can I just by looking at a cell tell what molecular pathway has been used? And that's a little bit challenging because I just showed you morphologically these two types, these cell types look different, but sometimes they're overlapping, and sometimes, in fact, you can have programmed necrosis even. So it looks like it's unprogrammed, but in fact it is. And therefore, morphology is a first, but normally what we do is we go and look at the biochemistry. We actually ask which molecules are involved, because the phenotype, what it looks like, is actually the result of the molecules that do their job. All right, so that leads to the question then, well, what do we know about the molecular basis of apoptosis? How do you actually get a cell to self-destruct, to cause suicide? And I'll show you 5 things that we know happen in the cell when it dies. So the first thing we know is that the DNA, the genomic blueprint, gets cleaved by a DNA called CAD. So if you think of our DNA, the blue thing here is the DNA. It goes on forever and forever. In each cell, we have 2 meters of this thing. The cell is 20 microns. We have 2 meters of this thing. And so we package it in these things called nucleosomes, and so these DNAs will cut here. And so that makes little strands of DNA, and these strands can be separated using a physical process called electrophoresis, and you can see what is called the DNA ladder. So this is DNA from 1 little ball. This is the DNA for 2 little balls, 3 little balls, 4 little balls. The DNA necrosis also gets pulled into small strings, but it's not specified, right? It's simply if you go and you shear the cell to bits, you'll break this thing apart, but it's going to be completely random. And so you can see biochemically that you get a clean cleavage here, and here you get just this messy cleavage. A second thing that we see is that the cell changes its outer surface. So our cells, think of them, I guess, like orange. They have a peel, and the outer part of the peel looks different from the inner part. Now, in our case, the peel is very, very thin. It's only 2 molecules wide. We have the outer leaflet, we call it, and the inner leaflet. And we have different molecules outside and inside. And that's useful because they have different functions. Now what happens when a cell dies is that some of these molecules that are normally only found inside suddenly go to the outside. And that's one of the signals that the system uses to say this cell is dying. So once I put my heart out on my sleeve, if you want, that means I'm about to die. You can easily see that on this movie. Here we have a cell, right? And what you will see is a red stain, a red surface appear. So we have a fluorescent protein in this bath, if you want. It's just floating around. And it would love to bind to a molecule that's on the inner leaflet. So it's hidden because it's inside and the red thing is outside. But what we'll do is once these things go outside, it will be able to bind and then the cell itself will become red. So let's try this here. So you can ignore the green part for right now. We'll discuss it in a second. So as the cell becomes small and shrinks, this is what it looks like when it dies. You can see that the complete surface becomes red. And again, the other thing you see is that it breaks up into little pieces. So the big cell is actually too big to eat. And so you have to make little tapas, little pieces that are easier to eat from the neighbors. All right. Now, this is good because it helps us get rid of the cell. The other thing that I wanted to show you here is focus now on the green. Within each of our cells, we have what are called mitochondria. We have a few hundred of these per cell, and this is where we make our energy. And these mitochondria are small units, and that's why you see the green in blips, because each one of these is one small unit. Now, when a cell dies, part of the proteins in these units are released into the sea of the cell. And you'll see that, and it's interestingly one of the first things that happen. You'll see that suddenly, before you see any other change in the cell, you'll see this thing become, we'll try again. You'll see like it becomes uniform. Within a few seconds, it becomes uniform green. You saw it. Anyway, what is this? No, I'm angry. Try this again. No, it doesn't work. Oh, no, yeah, it does work. All right. Hengartner just has to be careful. Gentle, gentle, gentle. There you go. See? Wow. So now it's all over the place. And right after that, the cell collapses. Forget it. All right. The last thing I want to tell you is that when a cell dies, we start having a disassembly team being activated, a set of what we call proteases, proteins that cleave other proteins, that will go and cleave a few hundred of our proteins. Every cell has thousands, probably 10,000 different proteins being expressed. A small fraction of those, but more than one or two, get cleaved very cleanly. It's not a degradation. They're not just destroyed. They're cleaved very precisely. All right. So these markers, these cleavage can be used extremely usefully in the clinic. And that's a case example here. So I told you that apoptosis is the way most of our chemotherapy agents work, right? So this now is an example where we're going to try to measure apoptosis directly to realize if a chemotherapy agent works. This is a patient, has a liver metastases, and the physician doesn't know which product to give, right? So he tries first this thing called AMP vinorelbine. And what you see is first that the tumor continues to grow. This is in fact PSA, prostate-specific antigen. So this is a prostate cancer with liver metastases. And this tumor grows, and so there's more and more and more of this thing. And what you see is that if you measure apoptosis, it stays. So even though you have cycles after cycles of chemotherapy, the cell doesn't respond. It doesn't respond to this apoptotic signal. They tried in a second different drug, and what you can see is every cycle, whenever you give the drug, you have this peak in cell death, poke, 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 and now suddenly the level of PSA, so a marker of the tumor, goes down. And if you try to measure this metastasis, you can see that it was 2.7, then it was 3.9, it goes down to 2. So here, this is really good, because if you wait for this, you see what it happens. You've lost 7 cycles. You lost like 3 months of chemotherapy, right? And so in this time, the tumor grew and it might spread more. Whereas here, you can see within 1 cycle, you know if it works or not. And so this is a really helpful marker to realize very rapidly whether your drug treatment works or not. All right. I'm looking at the time. We'll very briefly tell you a little bit now about 3 classes of molecules that are important to kill these cells, and don't try to get all the details, but just try to get the big picture. All right. So the first class of molecules we need are these this assembly crew. These are known as caspases. They're proteases. So again, they cleave specifically proteins, and they're known as caspases because they cleave after one particular amino acid known as aspartate. These caspases are in all cells all the time, but they're not active, right? So if you want, the gun is always in our pockets and it's loaded all the time. But somehow we need to activate the trigger. And that is done by having these proteins come together. So it turns out that these caspases, when they're all alone, floating around in the cell, are not active. But when they dimerize, when they come together, when you have two of them at the same time at the same place, then they're active and then they can kill the cell. And so one of the big questions was, well, how do you control this? How do you control these things coming together to become active? And so for that, you need a second group of proteins known as the death domain proteins. So biologists like to be dramatic. So we have the death domain and the death effector domain and things like that. And these are like Velcro. So a protein that has one of these death domains will bind to another protein that has the same death domain. And again, we have modular Velcros. So these guys will only bind to other guys with the same thing. These guys will bind to other guys with this domain. These guys will bind to other guys with this domain. And so it gives you this modular Velcro sets where having two proteins with these things allow them to come together. All right, so how can we use this to kill cells? I'll show you two examples. One of them is the mechanism that is used by our immune system to very rapidly kill a cell that is, for example, infected by a virus. And so this functions very rapidly because it directly activates the caspase. So it turns out that on all our cells, we have death receptors. These are there, and if they get the signal from outside saying, please die, they will. How does this work? The immune cells, the killer cells that protect our bodies, have these ligands which they can activate, and if they are active, they will bind this receptor. Now it turns out this protein on the surface of the cell has some of these death domains. When these aggregate, when they cluster together, you'll take out of the solution, out of the cytosol, out of the system in the cell, you'll take an adapter molecule known as FAD. Please don't remember the names. This thing has another of these Velcros. So the first set of Velcro allows you to bring this protein here. Then you have a second set of Velcro. And the second set of Velcro brings one of these caspases, one of these deconstruction machines, to the surface. And because now you have a lot of these together, they become active. And so they go, it's a mechanism we call induced proximity, and they activate the next set of these caspases, and then you have these hundreds of things being dismounted, right? The second way in which we can do that is the way that is normally used. It's slower, and it involves this mitochondria I told you about. So this takes another of these caspases, caspase 9. It again has one of these Velcro. This is now one of these cards. And again, you need to bring these caspases together. And so for that, we use an adapter molecule. It's called APAF1. Don't ask for the name. It has no meaning. Now, this thing here is also floating in a cell, and it needs to aggregate. And that it does by a sensor domain here that binds to a thing called cytochrome C. And this cytochrome C is exactly one of these things that are found in mitochondria. So how does this work? So we have these power units, these power plants, mitochondria, and they normally do their things. And when a cell needs to die, suddenly the power plant releases this unit from the power plant to go into the city and say, well, you need to die if you want. You assemble these units of death, and then the cell kills itself. Now, the weird thing about this is, and we don't understand it to this day, so I cannot explain, is the cytochrome C is here. So we have this membrane again. And this membrane, that's like a huge wall. Big things simply don't pass it normally. And so one of the questions is, how does this thing ever get out? Because it's too big to simply diffuse. You somehow need to rip open this. Now, we still don't know how it happens, but I can tell you the guys who do it, right? And these are a bunch of proteins known as the BCL2 family, and they're called BCL2. B-cell means B-cell lymphoma. This is one of these cancer genes. And what happens is that when you have this thing activated, the cell can die, it accumulates damage, and it kills itself. So, we have cytochrome C, it needs to get out. This happens by having these active backs on this membrane. This is one of these BCL2 family members. Now again, this thing is not around active normally, it's inactive. So what do we need? We need a signal. So imagine again DNA damage, somehow activates. I told you about these sensors. These BH3 domains are actually the sensors that will activate BAX and kill it. Now you can imagine that some cells are very valuable to us and some cells we are happy to get rid of. And so for the valuable cells, we need to make sure that this doesn't happen too easily. And so because of that, we also have BCL2 family members that prevent this from happening too easily. So it's like a shield, right? And so as long as I have some of this shield, I can mop up, a sponge if you want, I can mop up the system. Because this shield is very effective, if you really need to kill the cell, you'll use more of these sensors to get rid of the shield. Now this sounds very complicated. Let me give you an analogy, right? So what normally happens is you have this stress, you activate these the activators, they go to the executioner, and you have this cytochrome C release. You can block this by having these survival molecules, and you can inhibit this by having that. All right, so let's take the same thing, but simply call it organized crime. I come from New York City. So you have the stress, that's Mr. Big. Mr. Big wants something bad to happen, right? So what does he do? He hires henchmen. The henchmen themselves don't do anything, but they'll go and they'll get hitmen, and the hitmen make sure that something bad happens, cytochrome C gets out of the cell, so the victim dies, right? Now, you don't want this to happen too easily, so what do you have? Well, you have cops. The cops make sure that they can't really touch Mr. Big, right? So what they do is they inhibit the henchmen, they find the henchmen and the hitmen, and they block them. Now, if Mr. Big really wants this victim to die, he needs to somehow take the cops away. And how do you do that? Well, you have donuts. The donuts go and get the cop. And so they're busy with the donuts, and then this can go. The same thing happens at the molecular level when our cells undergo apoptosis. All right, the last slide. I told you that this is central to how we die. And because of that, now there's a whole bunch of new molecules that have been developed, which are now in clinical trials, where we try selectively to do what normally happens. So if you have a small molecule that acts like these donuts here, you can imagine that then this would happen more readily, right? And if you know, and it turns out there's many different one of these, and so you can make one that looks like a pink donut, and the other one is a chocolate donut, and so forth. And so depending on the cancer, you know which kind of donuts you need. And so that allows you to have selectivity, and that's basically the way in which biologists and physicians now try to go about it. There's nothing that's in the clinic yet, but there's a lot of things that are in clinical trials and looking quite promising. And so perhaps 10 or 15 years after what this prediction said, we will get, in fact, this huge market. With that, I close. What I told you today is that active cell suicide, called apoptosis, is a mechanism that is used in our bodies to remove cells that are either in excess, in the way, or potentially dangerous. And I showed you that the effects in apoptosis are observed in many human diseases. I showed you a teeny bit that it's a complex molecular dance that happens. I gave you an analogy that perhaps you will remember that helps you realize that it's not that trivial and that one needs to finely tune this. And of course, it needs to be finely tuned. It's a matter of life and death, ladies and gentlemen. And finally, I told you that this is clinically very relevant because many human diseases, cancer, but also stroke, heart attacks, have an effect involving apoptosis, that we can use it both for biomarkers, but also for therapy, and that therefore it's a very attractive approach to treat human diseases, in particular cancer. And with this I close, and I thank for your attention.
A lot of people go to dangerous places, like places where there's higher natural radiation, to get more healthy. And I think, if I understand correctly, then this is exactly what happens. This dangerous environment, the more radiation they take in, damages more cells, it creates more apoptosis, and the body starts to basically refresh itself.
Rejuvenate.
Yeah.
Yeah, I know people who take ozone therapy for the same reason.
That's part of it. It might be part of it. The other thing that happens actually is if you get relatively low doses of additional radiation, your body actually becomes resistant, increases its defense mechanism. And then if you get a bad dose, you're much more resistant than if you hadn't been primed. And so priming also helps you for the bad case. So for example, if you're going to suffer from hypoxia, if you're going to have a stroke, if you could before the stroke just hold your breath for a little while, for a little things, Or again, when you go to Everest, you first go to base camp, and then you go to camp 1, and this allows your body to adapt to this system. Yes.
So obviously you need to have a multi-cell animal for this to work. So from how many cells actually you see this apoptosis popping up? I mean, if you have a unicell animal, probably it will not kill itself.
You'll be shocked. You'll be shocked. So what we call apoptosis, this process here using these proteins, you only find in multicellular animals. So it turns out you have multicellular plants. They also have, I told you about the leaves, that is programmed cell death. It's in different molecular models. Now it turns out you have programmed cell death in unicellular organisms. And there's 2 examples I can give you. One is as soon as you have social unicellular organisms, where you kill yourself, but your neighbors who are genetically very similar might benefit from your death, it will happen, right? So it's like you sacrificing yourself for your kids, right? I mean, you're dead, but the genes live on. The second case we have is when we have parasites. So there are some very clever viruses that can infect the cell, not kill the cell, but stay with the cell. But then when things go bad, they want to get out. And so they can then kill the cell actively. Or we have examples where we have what we call plasmid addiction. So these are now parasitic genes, if you want, where the cell can try to get rid of the gene. But the gene consists of 2 parts, a toxin and an antitoxin. And if you lose the gene, you lose the possibility of making the antitoxin. And now this parasite is clever enough, it makes the toxin very stable, very long-lived. The antitoxin is very unstable, very short-lived. And so you need to continuously make the antitoxin. And as long as you lose that gene, the toxin survives, the antitoxin goes back, and you kill yourself. And so again, it's a way for this parasite to make sure that the cell can't get rid of it. And we assume, in fact, that this model, this thing, came from unicellular organisms initially. So no, that's the cool thing with biology. Anything you pretty much can imagine, you'll find out there.
2 questions. I got a question. And I was wondering, at some point in one slide, you listed a long list of, if I understood correctly, possible treatments to induce.
Oh yeah.
Yeah. But then I wonder how it's possible that you can, I mean, how much they have in common that completely...
Nothing. That's the problem. So again, but remember Woody Allen. So we have these neurotic cells. They check, am I too cold, am I too hot, am I too warm, am I too basic, am I too acidic, do I have not enough oxygen, too little oxygen, how my neighbors doing, are they okay, do they still love me? So there's so many different things that these cells feel, sense. And all you need to do is screw up one of these 20 sensors. And so that's why it works. So you can oxidize the cell, you can reduce the cell.
But then how is possible that cancer cell escape? I mean, it should be at a bottleneck where you can block all of them.
No, you can induce all of them. That's a problem. And again, you can kill every cancer that I've ever met. You can kill. The problem is if the cancer cell is more resistant than the next cell, normal cell, you'll kill the normal cell before the cancer cell. Right? So we need molecules that preferentially kill cancer cells, or 1,000 times better at killing a cancer cell than a normal cell. And then you have something interesting.
But when the body regulates sort of the cancer, how does the body mechanism detect the cancer cell from the non-cancer cell?
Mostly the immune system will do that. So the cancer cell normally has mutations and will start expressing things that regular cells will not express, and at least not at these levels. And so the immune cell will actually recognize the cancer as foreign, as not part of our body, and will attack it. And what does the cancer cell do? It tries to hide its difference and will try to inhibit the immune system. I mean, it's a battle, it's a race inside the body between the immune system and the cancer system.
And maybe the other thing is we only detect the successful cancer. I mean, our body is killing cancer all the time.
Exactly.
While we're sitting here.
Exactly.
So millions of types of cancer in your body are getting rid of right now. These are the types of cancer that the body can detect, but only those which it cannot detect. So while those things that fit in this very narrow bandwidth where they say, I'm one of you, please give me oxygen and food and everything so I can grow.
again, what's surprising is not how frequently we get cancer, it's how rarely we get cancer.